Well, welcome back to this session. This one to me is a very important one because for years you were led by a belief that said genes turn on and off and control the character of your life. Then you put that into context. As far as we know, you didn't pick the genes you came with. And then you can't change the genes if you don't like your characteristics. And then, of course, at the end, if they turn on and off without you being involved, then by definition, you become a victim of your heredity and everyone is believing the fact that genes are controlling their life. Well, this is 100% false. I'm so glad to say that because there's a new understanding from the basis of what we talk about as epigenetics, how things really work. Well, let me go back to a beginning part here, and that is I was teaching medical students about how genes control life, yet in my lab I was doing the research that we described earlier on cloning stem cells, revealing how the culture environment really was controlling the genes. And the significance about all that is that all of a sudden it says, well, it's the chemistry of the environment in your body that actually selects your genes. The genes are not activating on their own. And then I said, well, how does all this work? Well, this is where my colleagues got upset with me because I could show them and predict and say, look, I put this medium in with the cells and the cells will either form muscle or bone or fat based on which medium I put in the culture dish. And they go, yeah, because they were all involved with genetics at that time in 1967. And so basically I was like the weirdo in the room, like, what do you mean it's not genes? And I said, it's not genes, it's environment doing this. And the big question that they came up was, how does that work? And in fact, I really didn't have an understanding of how it worked because I could observe that it worked by changing the medium. I changed the fate of the cells. How did it work? Well, that led me into an investigation of how do those environmental signals come from the environment and go inside and adjust the genetics. Well, to start with, here's a cartoon version of cells in a petri dish, and what I want to illustrate with it is that if I add my culture medium with specific chemicals or signals in it, remember signals activate proteins. When I add these signals to the culture medium, the signals bind to the surface of the cell, the membrane, the skin of the cell. But then what happens is the signal is translated and sent inside the cell. And when the signal goes in, remember, signal plus protein equals behavior. And so it's the environmental signal that's engaging the behavior of the cell. And then more importantly is this, in order for this to work, you have to have the proteins in the cell. So the big question is, what happens if a signal shows up, but there are no proteins in the cell to respond to it at that time? That's when then the signal from the environment goes into the nucleus of the cell and activates the reading of the cell. So the signals from the environment control the behavior, and the signals from the environment control the genetics. Well, this led to a new science, a later science than when I started, but a science that made sense of what I was showing, and that science is called signal transduction. Big complicated name. Basically what it says is the study of how an environmental signal controls the biology, signal transduction. And a subset of signal transduction is specifically when the signal goes from the environment into the nucleus and activates the genes. Well, that's a special name. That's a subdivision of signal transduction. It's called epigenetics. Epi, above the genes. And I say, what's above the genes? And the answer is the signal from the environment is controlling genetic activity. So this was the science that later evolved. I was there trying to figure out how it worked because, of course, the big question was, okay, Bruce, we see your results, but explain it. Well, I had to understand something, and the understanding was this. The membrane was more than just a skin. The membrane was an information processor. So I say, where's the brain of the cell? Because the cell is responding to the world. It's not like a pinball bouncing all around. No, it is controlled by what's going on in the environment. So I say, oh, then the membrane is the actual basis of what the brain of the cell is, the membrane, the skin of the cell. Interesting little side story about that is when you were an embryo, where did your nervous system come from? And the answer is the skin on the back of the embryo turns in and creates a spinal cord and a brain. So guess what? You and the cell, both of us, have our brain created from the environment through the skin. And of course that makes sense because the skin is the interface between the outer world and the inner world. So I said, how does it work? Well, when you look at the nature of the molecular structure of the skin, it is one of the simplest structures in biology. In the microscope, and it has to be an electron microscope because the skin is so thin you can't see it with a regular microscope, that in fact, biologists didn't even know cells had membranes until the late 1940s when the electron microscope was developed and they could see it. Well, in the microscope, all you see is a little dark light, dark layer, very thin layer over the surface, dark light, dark. And in biology, a very interesting concept was that the complexity of the function was related to the complexity of the structure. But when you looked at the cell membrane, it had the simplest structure of all, just dark light, dark layer. There was nothing to it more than that. So most scientists thought the skin of the cell was nothing more than like plastic wrap holding the cytoplasm together. But as I started to look at it, I said, so what's the nature of this membrane? Well, yes, there is a dark light, dark, as illustrated in this photograph of the molecular structure of the membrane. It's called a bilayer. It's like a mirror image. You can see molecules on the top and a mirror image of those molecules underneath. It's called a molecular bilayer. The molecules are called phospholipids. The center of the membrane, the dark light, dark, the light part, is actually where the lipids of the phospholipids are to be found. Lipids are like oil or butter, for example. And I say, what's the relevance of that? And the answer is very simply this. Water-dissolved elements that live in water, hydrophilic, water-loving elements, can come to the surface of the cell, but they can't go through the cell membrane because below that surface is a layer of fat, lipid, oil, which doesn't allow water-dissolved substances to come into the cell. So I said, well, this is a barrier. That's what everybody said it is. The membrane's a barrier. But what they couldn't see in the electron microscope is there were proteins built into that cell membrane. And there are two classes of proteins. One class of proteins has antennas on it, and that is called a receptor. The other class of proteins I want to describe is called a channel. And what is a channel? Well, a channel is a conduit, a channel like a pipe going through. I say, remember, proteins change their shape and respond to a signal. So when you look at a channel protein, at first the protein is closed. So there is no conduit. The channel is closed. But when a signal comes to that channel, the protein changes shape, opens up, and makes a tunnel. And that's how information can get in and out of the cell through these particular proteins called channels. So we have two different kinds of proteins built into the membrane, receptors and channels. Well, this system of input and output, receptors represent input, channel represents output, receptor represents stimulus, the channel represents response, and I go stimulus response. That's like a reflex arc. Signals come into the receptor, which is a protein, change the shape of that receptor, allow it to connect to the channel, which changes the shape of the channel, and then the channel opens up and the signal goes into the cell. So the cell membrane is an information processor. It takes environmental information and translates it into a biological signal via the receptor and the channel complex. So we have now an information mechanism built into the membrane. If we look at the surface of the cell, in this cartoon version of the surface, you can see there are proteins scattered all through the surface built into the membrane, and that these proteins have antennas. The antennas are actually sugar molecules, very complex molecules. And so these are called receptors. The sugar group is like the antenna that responds to the signals in the environment. And then when a signal is picked up, it causes the protein in the membrane to change shape, which allows it then to connect to the output, the channel. So I said, well, what does the surface actually represent? And the beautiful part is it's the equivalent of a keyboard, that the environmental signals type on the cell by activating the receptors, and then that activates a response that goes through the channel. So the environment is typing on the surface of the cell with its signals, and these signals are translated by the membrane into biological behavior. Well, that's easy to describe, but it's even easier to show how it works. Here's an animation showing like a cookie cutout of a piece of membrane. And in this piece of membrane, I show a receptor with an antenna and a channel. And you notice that the channel is closed. It's not open at all. And I say, well, this is a switch. I say, what do you mean? I say, when a signal comes, the receptor will be activated, which will then allow it to connect to the channel, which turns on the information going into the cell. It's sort of like a light switch, where you hit the switch and then you get a current flow. Well, the switch is the receptor and the current flow goes through the channel. So as we see this in live action, then we see the antennas looking for a signal. Now, what we're looking at is a small protein, a green protein. In this case, it's called a G protein or a processor. It's the one that connects the receptor. But if you look at it, the processor couldn't fit onto the protein before the signal. But once the signal showed up, guess what? It changed the shape of the processor and of the receptor, and that allowed the processor protein to connect from the receptor to the channel. And when that happens, as you can see, the information now can flow through the channel because the channel opens up and actually makes a conduit between the inside and the outside. So look at it again. Signal shows up, changes the receptor shape, allows it to couple to the processor, which then couples to the channel, changing the channel from a closed channel to an open channel. And that is how information is taken from the environment via the receptor, connecting it to the channel, and the channel opens up and sends a signal inside. That's a basic switch. And I say, well, what's the name of this switch? In biological terms, I would call it, by its accurate name, it's called a receptor effector integral membrane protein complex. And everybody goes, ooh, that sounds so good. And I go, it's Latin in the sense that science converts it into language that the average people don't understand what it is. Receptor effector integral membrane protein complexes. Yeah, what is that? Well, let me explain the nature of the switch using the definition of the function of each of these parts. So I say, what's the function of the receptor? Well, in the switch function, the receptor's function is to read an environmental signal. Now listen, these receptors are built into the skin of the cell. Again, let me remind you, your basic receptors are also built into the skin of the cell. Your receptors are what? Eyes, ears, nose, taste, touch, pain, temperature. I say, where are your receptors built into the skin? I go, this is exactly the same as in the cell membrane, and they're simplified receptors, but they do exactly what our complex ones do. Our receptors read the environment so we know what's going on. The cell's receptors read the environment so they know what's going on. So I say, then what's the function of the receptor? The function of the receptor represents simply awareness of the environment. What's going on in the world? The receptor can see, feel, smell, touch what's going on. And I said, well, then what's the function of the channel? I say, well, when activated, the channel sends a physical signal into the cell that activates the proteins or the genetic activity. So I say, then the function of the channel is to create a physical sensation that will travel into the cell and control the functions inside. So now I can say, well, let's define the switch this way. And I say, the switch represents awareness of the environment through a physical sensation. I go, wow, that's a simpler understanding of what a switch is, but I want to even simplify it more. Why? That definition is already in the dictionary. There's a word in the dictionary, the word is perception. And when we understand the nature of perception, guess what? It represents awareness of the elements of environment through physical sensation. I go, oh my God, the definition of perception, the definition of the switch, exactly the same. Point? The switches in our membranes are units of perception. And as such, then the perception that the cell experiences is translated to a switch, which turns on and off the functions of the cell. So a switch is perception. But interestingly, perceptions are how we interpret the world, how we see the world. So all of a sudden I go, well, are all of our perceptions accurate? Well, of course not. We have misperceptions. I go, why is this relevant? Well, a misperception will activate the cell just as much as a real perception does. And I say significance then is we could call the switch perception, but since it can be a true perception or a false perception, a more accurate interpretation of the switch is that it represents belief. It's whether you believe this or you believe that, that's what engages the switch and controls your biology. So our perceptions are controlling life. I go, yeah, but how does this affect the genes? Well, the interesting part is when you open up the nucleus of the cell, you can open up the nucleus and see there are what are called 23 chromosomes doubled, meaning 23 of the chromosomes in the nucleus came from your mother, 23 of the chromosomes in the nucleus come from your father, 46 chromosomes. Yeah, but what is a chromosome? Well, it's a unit of heredity. I say, yeah, but what is it? Well, a chromosome is made out of, guess what? 50% DNA and 50% protein. Ever since DNA was understood from the 1950s, scientists were so focused on the DNA, they didn't really look and care about what the proteins were doing in that chromosome. So they would isolate the chromosome, separate the DNA from the protein, keep the DNA, and throw away the protein. They did that for about 50 years until somebody said, wait a minute, 50% of the chromosome is protein. Maybe it's doing something. And all of a sudden they say, well, what is the protein doing? So we start to understand that what's going on is we didn't know before, but now we know. And I go, what do we know? A chromosome, let me just illustrate it like this. A chromosome has a core of DNA and a sleeve of protein surrounding it. And I go, so what does that represent? I said, well, look, let me illustrate it using my own arm. This is a representation of a chromosome. And I go, what does it represent? I say, there's a sleeve of protein covering up DNA. And I go, oh, so the DNA are the genes. I go, yeah, so let's say I take a magic marker and I write the genetic code for blue eyes onto my arm. This piece of DNA is the gene for blue eyes, let's say. I hold up my arm, I say, can you read the gene for blue eyes? And the answer, of course, I could read the genetic code right there. But I say, oh, no, what does it look like when it's inside the cell? Oh, inside the cell, the DNA is covered up by the sleeve. I say, can you read the gene for blue eyes? You say, no. I say, but if I need the gene for blue eyes, then how do I read it? And the answer is, remember, the sleeve is protein. Remember that when a signal from the environment binds to a protein, it changes the shape of the protein. So when a signal comes in that binds a protein on the surface of the chromosome, it will cause the sleeve to come off, change its shape, and then you could read the genetic code. So I go, why is this relevant? And the answer is, well, the gene didn't do anything. It was covered up. But to read it, you have to take the sleeve off. To take the sleeve off, what you had to do was actually then put a signal into the system that would cause the sleeve to change shape. Now let's illustrate this in an animation. And in this animation, you can see an example of a chromosome. It's covered by a layer of proteins, and inside is a core of DNA where the genes are. And at the beginning of each specific gene, there's a specialized protein called a regulatory protein. Regulatory, that's already an informative name, regulatory. I say, how does it work? I say, when a signal comes from the environment, it goes in the nucleus. It will complement the protein that it best fits. So the signal is very specific to identify which regulatory protein we want to activate. And when the signal comes in, it binds to the regulatory protein, causes that protein to change shape, the sleeve of protein pops off, and now the gene is exposed. Then I say, what happens then? Well, as we're going to see in this video right now, what you're going to see is first the signal comes into the nucleus. And then what it does is it finds the protein it can complement with, the regulatory protein. Once the signal binds to this regulatory protein, it causes the proteins to change shape. And when that happens, what do you think happens? The protein sleeve pops off. Now you can read the gene because there's the gene underneath that protein. So I said, well, then what happens? I said, well, another protein called a polymerase will come up, copy the DNA, and turn it into a Xerox version, so to speak, of that gene called RNA. And that is the future blueprint that's going to go into the cell, and that blueprint is going to be used to create the protein. Once the RNA is made, I don't need the gene anymore, so the proteins come back on and cover up the gene. So let's look at it one more time in a action. Here comes a signal. It's going to find the protein and complements it, bind to it, cause the protein to change shape, the sleeve pops off, the DNA is exposed, a protein comes up, copies the DNA, making RNA, which then goes into the cell and is used as the blueprint to make the protein. After the RNA is made, the sleeve pops back on, and that's how it worked. Well, if you just looked at that video, I could ask you a question. What did the gene do? Because in our history and what you've learned, oh, genes are activating and genes are turning things on and off. I go, if you look at the video, you recognize the gene didn't do anything. What it was a template. And what you had to do was expose that template with the environmental signal, so then we could copy the gene, make the RNA blueprint, and that goes into the cell and is used to make the protein. The point is simply this. The DNA had no action in this. The DNA didn't turn on, it didn't turn off, it didn't control anything. It was the signals from the environment and the proteins that were controlling the readout of that DNA. So if I take a piece of DNA, like in this illustration right here, out of the chromosome, and I can show you here's a strand of DNA, and there's a loop in here, and let's just say that loop is the gene, let's say the gene for blue eyes. Well, guess what? The total amount of DNA that actually is represented by genes is about 3% of the total DNA in your nucleus. So that all of a sudden says 3% make genes. What is 97% of the DNA doing? Well, the funny part is scientists, being inefficient as people are, said, oh, well, that must be called junk DNA. Why? There's no function to it. It's not a gene. And that means 97% of the DNA in your nucleus was called junk because they thought it wasn't functional. But we know something different quite now. We know that that DNA has protein modules, groups of proteins scattered along that DNA. And I say, what is the relevance of those? I say, those are the proteins that control the readout of the gene. There's a module of that DNA that it turns the gene on and off. There's another module that determines how much of that gene we want to make. There are other modules that are profoundly important. I say, what do? Well, they're the equivalent of scissors and tape. I go, what do you mean? Well, they take the gene blueprint that you just copied, and they can cut it up into pieces and then tape it together in a different sequence and make a different gene. So a single gene can be read in a way that I could cut it and alter it to make different blueprints from the same gene. Now, I didn't change the gene. I changed the readout of the gene, the so-called RNA. So the modules in the DNA are like the dials on a television set that you can turn it on, turn it off, change the color, the contrast, change the height, the width of the picture. You can change the readout. Does it change the gene? No, it only affects the RNA. So epigenetics comes in, and epigenetics are the signals that control these modules and control the on and off, the volume, and what's going to happen to that DNA strip as it's cut and pasted together. So epigenetics affects the modules as the equivalent of dials on a television set adjusting the picture. Some of the epigenetic activity is to affect the readout of the genes. I go, how does it do that? And I say, well, look, you get 2 copies of each gene, one from your mother, one from your father. And in virtually every part of the biology, except for the immune system and the nervous system, you only use one of the pair of genes. So one of the genes has to be shut down, and the other one is the one that's going to be read. I said, well, what's the action of shutting it down? I go, epigenetic mechanisms select one of the pair and put what is called a methyl group, a chemical group, onto this particular gene, which inactivates it, and therefore only one of the pair is being read. So epigenetics changes the dials, which adjust the readout, and epigenetics can also say which of the pair of chromosomes is going to be read in the development of this individual. Well, say, well, how does epigenetic changes work to change the readout? And I go, well, it's called epigenetic gene splicing. I go, what does that mean? I go, well, remember, we make a copy of the gene, and that's called RNA. So we call it actually messenger RNA. It's a copy of the gene. I say, so what? Well, let me give you an example. Let's say here's the messenger in RNA from what is called the reading gene. And I go, so what's happening here? I say, well, you made a copy of the gene, the reading gene, and here's a copy. It's called RNA, and it's a copy of the gene. But then I say, so what does epigenetics do? I said, well, like scissors and tape, you can cut that signal, paste it together in a different sequence, and change the word. So, for example, let's look at it this way. Here's the reading gene. I cut off the ing part. Now I have the read gene. Or I have the reading gene, cut off the re part, and have the ding gene. Or I cut off the re and the and I left up with the add gene. Or I could cut out the middle of the message and then paste it back together and make the ring gene. What you're seeing on the screen right now is 5 different versions of the readout of the same gene. Now, it didn't change the genes. It's only affecting the copy of the gene called the RNA, okay? And I go, so why is this relevant? Why is this relevant? Well, you're only looking at an RNA with, 7 letters in it. Just figure this, an RNA could have 1,000 letters in it. And if you have 1,000 letters, you can cut that message up in so many different ways and put it together in so many different ways. I say, well, guess what? Using epigenetics, one gene blueprint can create over 3,000 different proteins just on the nature of how it was cut and spliced together again. So all of a sudden, you're not reading the exact gene anymore. You're reading alterations of that gene by cutting up the messenger and pasting it back in different sequences, okay? Well, to illustrate the point, here are 2 mice. The mouse on the right is a normal-looking mouse compared to the one on the left. The one on the left has different color fur, it's obese, it has cardiovascular issues, it has diabetes, and this mouse is a mutant called the agouti mutant. This mouse will have a very short lifespan and die. And you look at it and compare it to the normal mouse. But here's the interesting part of this picture. That so-called normal mouse has the exact same agouti mutation as the one with the blonde fur. I say, so what's the difference? Well, during the development of the mouse, during a short period in the window of the development, in a short period of just a few hours, if the mother is fed elements such as folic acid, vitamin B12, and choline, it can change the readout of the mutant gene so that it's no longer a mutant gene. And all of a sudden I said, well, that's why, look at the mouse. It's totally normal. But it has a mutant gene in it. If it mates, it will pass that mutant gene on to its offspring. But in this particular life, this mouse is not experiencing the agouti mutant. Epigenetics, the epigenetic environmental signals through what the mother was fed, adjusted the readout of the gene and canceled the mutation aspect, and it results in a normal mouse. So, epigenetics can take a mutant gene and make a normal individual out of it, and more importantly, this one, it could take a normal gene and make a mutant gene out of it. And this is why it's so important, because most of us came with a perfectly adequate set of genes to have a normal life. But if your epigenetic environment, remember, it's the environmental signals that are controlling this. If the environment is not in harmony with you, then the result is you can take a normal gene, modify it, and then express a mutant gene, and then have some kind of illness or disease affect you, even though you had normal genes.